Hi everyone, my name is Emma Schaefer, and in this presentation, I will be walking through the metabolism pathways and specifically how energy is acquired from sugars to create nucleotides. So we start with glucose, which is converted to two pyruvate molecules through glycolysis through a series of 10 steps. Through this pathway, we use 2 ATP, but we gain 4, so there is a net gain of 2 ATP. We also get 2 NADH, which is equivalent to 5 ATP. Pyruvate moves on to a different pathway to generate more energy after this. Conversely, pyruvate can be used to create glucose in times of fasting or starvation through gluconeogenesis. Other sugars can also be broken down to glucose or glucose 6 phosphate in order to enter glycolysis and generate energy. Some examples are shown here, lactose and galactose. After glycolysis, pyruvate is converted by pyruvate dehydrogenase to acetyl-CoA. This generates 2 NADH or 5 ATP, and this acetyl-CoA then enters the citric acid cycle, which through a series of reactions generates 6 NADH, 2 FADH2, and 2 GTP. Additionally, amino acids can be used to generate citric acid cycle intermediates. Alanine can be converted to pyruvate. Aspartate can be converted to oxaloacetate, and glutamate can be converted to alpha-ketoglutarate. Also, the NH3 that is created from the breakdown of amino acids can be used with CO2 in the nucleotide synthesis process, which we'll talk about later. The NADH and FADH2 generated by the previous processes then enters the electron transport chain, where an electron is passed through different complexes with increasing reduction potentials until it is passed to the final acceptor O2 to form H2O. This process pumps protons from the mitochondrial matrix to the intermembrane space. This gradient then triggers ATP synthase, which generates ATP through a spinning gamma subunit. As it spins through the different ATP conformations, ATP is released from the open site. Each NADH from the electron transport chain generates 2.5 ATP, and each FADH2 generates 1.5 ATP. To create more energy in the form of acetyl-CoA entering the citric acid cycle, we can also break down fatty acids. The glucose 6-phosphate that was created as a glycolysis intermediate can also enter the pentose phosphate pathway. This pathway generates ribose 5-phosphate, which is used to create nucleotides. Ribose 5-phosphate is converted to PRPP by PRPP synthetase, which can then be used with CO2 and NH3 that we got from the breakdown of amino acids to create UMP and IMP. UMP is then converted to UTP, CTP, and TTP, the pyrimidine nucleotides, and IMP is converted to ATP and GTP, the purine nucleotides. ATP synthase generates the ATP needed to create nucleotides because ATP is necessary for many steps, such as the conversion of the monophosphates to triphosphates. So a summary of these pathways is shown here. Proteins, sugars, and fatty acids can be broken down to create energy in the form of ATP, which is then used to create nucleotides and regenerate ATP to use as the energy source for other biological processes.